Tools of Molecular Genetics, Module 1, Lecture 3. Mutation is defined as a change in DNA which affects the function or translation from the gene to the protein. So mutation, for some conditions, a common mutation may cause all or a large percentage of all cases. Example, in sickle cell, a single change from CAG to CTG changes the RNA, which changes from a normal glutamic acid to valine. But in many disorders, there's not a single common mutation. There might be many that can result in a change in the gene. But let's start with a simple thing, sickle cell anemia. If only life was that easy for me, that a single nucleotide change, a single polymorphism, going from one letter to another, changes the amino acid, which also changes the function of the hemoglobin. A restriction enzyme is something that which we've used in attempts to determine gene changes. And basically, restriction enzymes are enzymes that cut DNA at specific places, and they use what's called palindromes. So they show this, they'll look for the sequence GAATTC, and then it will cut. There are over 3,500 different restriction enzymes that we can use, and we choose the specific restriction enzymes based on what we're looking for. So, methods to determine gene function: DNA, RNA, and protein. So we have our DNA sequence. We have to make a copy of it. We make our RNA transcript. We then slice out our introns, and we put a poly A tail and a cap on the other side, and now it's capable of being released from the nucleus into the cytoplasm. In the cytoplasm, it attaches to the ribosomes, and this transcript is used as a template to make a protein. Southern blotting looks for changes in the DNA sequence. Northern blotting looks for changes in the RNA, and Western blotting looks for the protein product. And in different situations, we may need these modalities, but you need to understand Southern looks for DNA, Northern looks for RNA, and Western looks for the end product, which is the protein. So Southern blotting, we've had around for probably over 50 years. We cut out DNA into small pieces using these restriction fragments, and then we separate them based on size. We then take it from the agarose gel and put it onto filter paper and use tags to determine where that particular thing is. We used to use radioactivity and x-rays, and we've gotten to fluorescent tags because it's a little bit dangerous. Northern blotting, this allows us to determine the abundance of RNA transcripts. And in cancer, this is actually very important because there are certain genes that are over-transcribed. And so we can determine in a situation of cancer how much RNA transcripts there are. And then Western blotting, this tells us if the protein is there. And this is actually the way we used to, we don't have to as much anymore, look for something called Duchenne's muscular dystrophy, which we'll talk about in future cases, that the dystrophin protein, this is what it should look like, and we have a different band in what's called Becker's, but we have nothing in the Duchenne's. So the band is a little bit different. Here's a normal dystrophin protein. Here is a smaller but at least functional dystrophin protein in Becker's, but we have no protein in Duchenne's. And this is actually the method we used to use to determine if somebody had a Duchenne's muscular dystrophy. So, as I said, sickle cell anemia, in a certain sense, it's nice because it's a single change to a signal gene. So, one way we can do a Southern is we have our patient's blood, and we cut it with a restriction enzyme known as MST1. In normal individuals, it cuts in the middle, so we get two small fragments. But when you have sickle cell, it only cuts here, so you'll get one large fragment. So we get two fragments here, but because this changed from CAG to CTG, we don't get this cut. So an individual with sickle cell, they'll have a fragment that migrates much slower because it's a larger piece. Whereas in an individual with normal hemoglobin, we get two fragments. So an individual that has just normal hemoglobin, you'll just see the two fragments. An individual that's a carrier, you'll see the normal hemoglobin and the sickle. So you can differentiate between having completely normal hemoglobin, being a carrier, and an individual that's affected will only exhibit this single large fragment. So we can even, here's a family history, mom and dad are both carriers, and you can see the sickle cell and the normal hemoglobin. This first child just has normal hemoglobin, the second child just has sickle, and the third is a carrier. So mutation screening, if only life was as easy as sickle cell anemia, in cystic fibrosis, there's over 1,400 different changes in the CFTR gene. And some are unique to families. These include missense mutations, frameshift mutations, splicing, and nonsense. Delta F508 is the most common. So, sometimes we look for common mutations that may represent a significant portion. So here we have before and after carrier screening. So we're looking for common mutations. If you're Ashkenazi Jewish, the pickup rate is 94%. So your risk of being a carrier of cystic fibrosis before screening is 1 in 24. But with a carrier screening of 94%, if you didn't have one of those mutations, now your risk drops to 1 in 400. So it goes from 1 in 24 to 1 in 400. Non-Hispanic Caucasians, the pretty good pickup rate, 88%, but it's certainly not the 94. The risk before carrier screening based on population is 1 in 25, but because this is only 88%, we're now the negative, it's now only 1 in 208. Hispanics, the detection rate's only 72, but fortunately you have far less common to have, the risk of being a carrier is only 1 in 46, but after carrier screening for the common mutation, now you've only dropped to 1 in 64. African Americans, again, 65%, even less common, but 1 in 86, 186, and Asian Americans, only 50% are picked up by carrier screening, and even though the risk is a fourth of that of Ashkenazi Jews, after screening, it's only 1 in 182. So by looking just for common mutations, if you have a very high detection rate, you can significantly drop your post-testing risk. Whereas if you're in a population that the carrier pickup is so low, you're not really changing your risk significantly. And so after carrier screening of a negative carrier screening, actually the individual, the Ashkenazi Jewish population, is at the lowest risk, even though they were at the highest risk before you started the carrier screening. So for cystic fibrosis, or CF, screening panels are available, which again can test for the common mutations, but negative results may not significantly decrease the risk, especially if they're not in a population that the pickup rate is high. So it's important to understand these concepts. Mutation screening benefits is quick, it's cheap, it can decrease the risk. But again, and if you're in a high-risk population and you have a very high pickup rate, it's good. But if you're in a population that you only pick up 50%, it doesn't really change your risk all that much. So limitations, it doesn't tell you about rare mutations. You're only looking for common mutations. And again, negative testing just means that we didn't find anything. It doesn't for certain mean that the individual is not a carrier. Epigenetics is a whole new area of genetics that grows on a daily basis. We used to think, you look at the DNA if there's a change in the sequence. But what we've realized, and from that earlier slide, you have to have your normal sequence, you have to be able to make a piece of RNA, and then that RNA needs to be used as a template. And any of those steps that don't work, you have a problem. In epigenetics, certain genes can be covered with called these methyl groups. And when they're covered with methyl groups, they do not get transcribed. Therefore, the gene product is not made. So if you sequence the DNA, like with a southern, it will be normal. But you may not see any RNA transcripts or any protein transcripts. So again, methylation is the addition of AOH to the fifth position of cytosine. And this results in the gene not being transcribed. Whole genome or whole exome sequencing. So first I want to go over the single nucleotide polymorphin or SNP. Base pairs from one individual to another is 99% the same. Everyone has differences in their genetic code, however, that 0.01%. And so if at this locus, this first individual has an A, the second individual has a G, and the third individual has a T, this is known as a single nucleotide polymorphism. This may or may not be in a gene. Less than, only like 20 or 30% of our chromosome regions even codes for stuff. There's a lot of filler in between. So this may or may not have anything to do with a gene. And even if it does have to do with a gene, it may not change the amino acid that's put in. So the genome is the entire set of genetic instructions, including the 23 chromosomes and the mitochondria, representing about 3.1 billion base pairs. The exome is just a portion of the coding sequence. So if you look at it, it's just like maybe a slice of the apple. The use of next generation sequencing can be used to identify genetic causes of disease when specific syndromes not known. It's more efficient and affordable when you have a long list of possible genes, and it can identify a number of different mutations in cancer cells. So this is really growing in literally on the daily basis of its use in cancer. And RNA sequencing is also important. The benefit of exome, it reduces costs while focusing versus on the most likely genes. This is actually just a graph of showing the cost of DNA over time. It used to cost $10,000 for megabase of DNA sequence, and now it's pennies. And this has allowed us to do so much more. So next generation is also called massive parallel sequencing. It uses capillary electrophoresis and Sanger sequencing. The Human Genome Project took over 10 years and cost $3 billion. Fluorescent signals were used to determine the sequence in Sanger sequencing. So the, but it's done rapidly. So samples are run in a triad, the mother, father, and the child, in the case of pediatric situations, or it could be an adult. But having the parents is important because it allows us, we have so much DNA, we can, if we have, there are 3 answers we can get when doing genetic testing: yes, no, and I'm not sure what that means. And when we're looking at common genes like this CF gene or sickle cell gene, we've studied this for so long, we know exactly what that means. But some of the rare genes, a change doesn't necessarily mean that it doesn't work. So this helps us to determine that. So here we have the difference between massive parallel and Sanger, basic gene by gene sequencing is, in a gene by gene sequence, you're amplifying or you're analyzing a single sequence of DNA, whereas this process allows us to amplify the entire genome at the same time and then test it. And what we do is, so we amplify the entire genome and then we sequence it using different fluorescent tags for the different base pairs, and then we analyze where these tags are through mass spec. And so this is primarily done with a computer. It's not like nobody counts these like reds and greens. And so here's a video on massive parallel
Most interesting pathologist in the world, and I would like to talk to you about the next generation sequencing. The process begins with genomic DNA, but the DNA is far too long to be sequenced, so it must be broken into smaller pieces. This is done by a on a pulse, so we will need a sound so unpleasant it can actually shatter DNA. Ah, thank you very much, Justin. Now, the DNA must be split into two strands, that is, denatured. It is then hybridized to a surface, either a bead or a solid plate. Now, I do not wish to favor one platform over another, so instead, we will hybridize to Chuck Norris's beard. It's a spherical Chuck Norris, but still, sexy nonetheless. The DNA fragment is then amplified in either an emulsion PCR reaction or an isothermal bridge amplification reaction. Yes, these are very, very big words indeed, but just watch the video. Here is some music for you. And now at last we are left with a clonal cluster of DNA. The DNA is then sequenced together, in this case with fluorescent nucleotides. As they are added, they emit light of a certain color so that you know which nucleotide was added. Now, of course, there are other ways of doing this, such as monitoring current. But you must agree, this is the most pleasing to the eye. Ah, very beautiful.
Okay, we're going to try to move to the next slide here, which may or may not work. Let's see, we'll go back. Okay, so we're almost done. So, there is concern that a child has muscular dystrophy. A 6-year-old has calf hypertrophy, difficulty walking upstairs. A molecular study that can be used is, and the correct answer is Western blotting. So this concludes this module on molecular genetics.